The broadcast is now starting. All attendees are in listen-only mode.
Hello everybody, I'm Donald Murphy, a member of the marketing team here at PacBio, and I want to welcome everybody to the PacBio webinar series. Today's topic focuses on taking a closer look at smart sequencing and the Iso-Seq method, which allows you to generate full-length cDNA sequences with no required assembly, and our speakers are going to get into that in a little more detail in just a moment. So it's my pleasure today to introduce today's speakers, who will present in the following order. My colleague Elizabeth Tseng, who is a principal scientist here at PacBio, followed by Gloria Shenkman, who is a research fellow at the Dana-Farber Cancer Institute and Harvard Medical School. The presentation portion of the webinar will be followed by a question and answers session. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee dashboard. We will be recording this webinar and making it available for download in the next few days, so please keep an eye out for a follow-up email with a link to the recording. So with that, we'll get started, and welcome, Liz.
Thanks. Good morning, everyone. My name is Liz, and I'm a scientist at PacBio. Today, I'm going to talk to you about the Iso-Seq method, which is PacBio's offering for full-length transcript sequencing. So what is Iso-Seq? Traditional RNA-seq is done by fragmenting cDNA and then sequencing the fragmented reads with paired-end sequencing. The problem with this is that in order to identify the full-length isoforms, it becomes an assembly problem, but this is computationally challenging and sometimes intractable. In contrast, the Iso-Seq method produces full-length cDNA, and using the PacBio long accurate reads can sequence the full transcript. No assembly is required. Briefly touching upon smart sequencing, the characteristics of PacBio smart sequencing are we have very long and accurate reads, more than 99% accurate, in fact, for most of the Iso-Seq reads. And I'll show later how this is useful for many applications. We also have minimal GC bias. And the Iso-Seq method on the PacBio systems is a complete solution from sample prep to analysis. The library prep and sequencing and analysis can be done in one day each. With the Iso-Seq Express Kit, a total input RNA is recommended to be between 60 and 300 nanograms. This produces full-length cDNA and can support multiplexing of different tissues or samples. On the Sequel II system, a single SMRT Cell ALM is sufficient to exhaustively characterize the whole human transcriptome. Typically, a single SMRT Cell would produce up to 4 million full-length reads. Shown above in the table here are two human samples we've released recently, the UHR and the Alzheimer brain sample. Both samples produced over 4 million full-length reads, producing 16,000 to 17,000 unique genes and over 160,000 unique transcripts. The bioinformatics solution is also complete for Iso-Seq. Starting from the sequencing data, the Iso-Seq application in the official software SmartLink produces high-quality, full-length transcript sequences that can then be mapped back to the genome. Later, I will introduce the tool Scottie, which is available on GitHub, that can be used to classify full-length transcripts against annotations such as the GenCode. So Scottie, or Scottie 2, is a tool that I've developed and is available on GitHub for classifying full-length transcripts. The inputs are the Iso-Seq transcripts, the genome, annotations such as GenCode, and orthogonal data such as matching RNA-seq data, CagePeak, Poly-A motif list, and Intropolis, which is a repository of public RNA-seq junction databases. The output of Scottie is a classification report of every transcript against the reference database. Further, there are reports on every individual junction and graphics. Another feature that is part of SCONTI2 that I won't go into detail about is that using the junction and the classification report as well as orthogonal data, SCONTI2 can further filter away cDNA artifacts. If you want to know more about SCONTI, you can follow the link here. SCONTI2 should be used as a quality control tool. It is used for classification of isoforms against known transcripts, classification of junctions, identification and removal of cDNA artifacts, creating orthogonal supporting evidence for full length, and functionality. What SCONTI should not be used for is fixing issues with other parts of the analysis pipeline, such as aligner issues, low quality sequences, and multi-mapping. Here I am going to show the major classifications of transcripts that is used in the SCONTI2 nomenclature. Given a reference transcript, an Iso-Seq transcript is classified as a full splice match, or FSM, if it matches the reference perfectly. This means it has the same number of exons as the reference and each junction is precisely matching to the reference. If it's an incomplete splice match, or ISM, it means that all the existing exons of the Iso-Seq transcript matches the reference perfectly, except that it's missing one or more 5' exons. These could either be 5' degraded products or real alternative start sites. There are two categories of novel isoforms. Novel in catalog means it's a novel isoform using a combination of known junctions. Novel not in catalog means it's a novel isoform using at least one novel splice donor or acceptor site. Here I'm going to show the characteristics of Iso-Seq on the Sequel2 system using a recently released public data set, the Alzheimer brain on SMRT Cell 1M. This data set generated over 162,000 transcripts ranging from 80 bases to 14 kilobases with a mean of 3.3 kilobases. This is the breakdown that SCONTI2 provided against the GenCode 31 database. The majority of the genes are known, however, a significant portion of the transcript isoforms are novel. When we further break it down according to the classification that I've just shown you, we can see that the breakdown is about 20% of them are full splice matches that are perfect matches to the database. A smaller percentage of them are incomplete matches, and then a significant portion of novel isoforms using known or novel junctions. This is the categorization after SCONTI2 filtering, so we see very few of antisense and intergenic reads. To further provide evidence that the transcripts are full length, we matched to the Phantom5 cage-peak data. We see that for the full splice matches, the FSMs, 70% of the transcripts fall within 50 bases of an annotated cage-peak site. Even for the novel isoforms, a good portion of them also are close to a cage-peak. On the 3' end, a good portion of all of the transcripts have a poly-A motif detected within 50 bases upstream of the 3' end. In the next two slides, I will show you examples of Iso-Seq transcripts from the Alzheimer dataset. Shown here above in red are the Iso-Seq transcripts. Below is the GenCode annotation for the PLD3 gene. We see that with the Iso-Seq transcripts, we see alternative start and end sites, intron retention, and exon skipping events. In the second example, we're showing the PSEN2 gene, and interestingly, for two of the Iso-Seq transcripts, we see transcriptional read-through of the PSEN2 gene to the downstream COQA8 gene. Finally, the data is also highly accurate. Shown here is a histogram of the Iso-Seq transcripts mapped back to the HT38 reference genome. 90.5% of the transcripts matched to the HT38 genome with 100% identity. In fact, more than 99% of the sequences are more than 99% accurate. This high accuracy allows us to do direct open reading frame prediction. Shown here is the breakdown of the different categories again. However, shaded in the darker color for each category is the proportion of transcripts in that category that are predicted to have an open reading frame of longer than 50 amino acids. We see that for the novel isoforms such as the NSC, the majority of the transcripts are still predicted to be coding. To summarize, Iso-Seq on the Sequel2 system can be efficiently done within 3 days. I'd like to spend the last few minutes talking about who is using Iso-Seq. The first case I'm going to present is a targeted Iso-Seq study on the MYBPC3 gene for hypertrophic cardiomyopathy. This is the case of a 21-year-old female patient with severe hypertrophic cardiomyopathy. The HCM panel identified a single base mutation in the MYBPC3 gene that is expected to affect splicing between exon 19 and 20. When the authors used targeted Iso-Seq, they indeed identified an alternative isoform shown here, AS1.1 on the left side, that skips exon 20. When compared with other control and patient samples shown here on the right, we see that AS1.1 is only expressed in the study patient. However, in addition to AS1.1, other alternative splice patterns, AS1.2 through AS1.11, are also discovered. The alternative isoforms exhibit multiple different alternative splicing events such as intron retention and shifted splice acceptor sites. Because the reads were highly accurate, the authors were able to manually phase and assign each transcript to the 2 different alleles. Shown here on the right, the canonical MYBPC3 isoform is expressed on the wild-type allele, whereas all the alternative splice forms, AS1.1 through AS1.11, expressed exclusively on the spliced variant. To summarize, Iso-Seq identified a patient-specific alternative splice variant linked to the mutation that is not expressed on the wild-type. In the second example, the authors applied whole transcriptome sequencing to look for fusion transcripts in BL. In B-cell development, one IGH allele is silenced. The IGH DUX4 translocation is a subtype of BL that accounts for 7% of the pediatric cases. However, DUX4 is particularly difficult to sequence with short reads because it is in a GC-rich region. Using whole transcriptome Iso-Seq on a BL cell line, the authors identify functional IGH transcription, antisense DUX4, and also the IGH DUX4 fusion. Once again, using the highly accurate reads, the authors were able to manually assign the transcripts to the different alleles. Analysis showed that the functional IGH comes exclusively from the wild type, whereas the antisense DUX4 comes exclusively from the fusion allele, showing that the translocation comes from the silenced IGH allele. Based on the analysis, the authors provided this hypothesis that activation of IGH DUX4 is toxic due to overexpression of DUX4. Hence, during early B-cell development, only the wild-type IGH allele is viable. I have presented you 2 human biomedical cases of Iso-Seq. However, Iso-Seq has been widely applied for plant and animal, especially for genome annotation. I'm just going to highlight 3 specific cases here. Recently, the authors from USDA and Australia have presented a case of the Brahman Angus hybrid F1 cattle. Using Iso-Seq, they were able to identify allele-specific and tissue-specific isoform expression. In a recent webinar, another set of authors used Iso-Seq to identify tissue-specific transcripts associated with THC and CBD synthesis. And finally, authors from Washington State University recently presented at the PacBio North America UGM meeting on applying Iso-Seq to grizzly bears. The goal was to find tissue-specific alternative splicing related to the difference between hibernation and active state. This concludes my portion of the talk. I will now hand the presentation to Gloria.
Thank you, Liz, for the nice introduction. So for everyone out there, my name is Gloria Shenkman, and I'm a research fellow at the Center for Cancer Systems Biology at Dana-Farber Cancer Institute and Harvard Medical School. And I'm happy to share with you our experiences using Iso-Seq to characterize full-length transcriptomes. So in particular, I want to make a couple of points today. Mostly with the new platform, how we're reaching much higher coverage and saturation for complex human transcriptomes, and thus discovering the full range of isoform diversity. I'll touch on some potential for isoform quantification, which I believe is a very promising application, and then end with some experimental design considerations, which includes when to use targeted versus bulk sequencing. So to start, what we most recently analyzed are some breast cancer and melanoma cell lines. And so the way our experimental design went is as shown. So we had 5 breast cancer related cell lines and 8 melanoma related samples. For each of these samples, when we converted the RNA to cDNA, we barcoded. So each sample has a sample-specific barcode, and thus we could pool everything into one library for sequencing on the Sequel II system. So these are some sequencing run statistics, in particular the raw reads. So again, each of these pools of cancer samples were run on a single 8M chip. On average, our base yield was about 300 gigabases, and we obtained about 6 million polymerase reads. And so this is fairly typical, I believe, for this particular configuration. To the right, I'm showing you the polymerase read length distribution for our breast cancer samples. And so you can see that many of the reads are very, very long. And on average, the raw read length is about 20 to 30 kilobases. And so given that the transcript is from human samples are about 2 kb, that molecule is being sequenced many times. And then we can cluster or average the nucleotide sequences so that we have much higher accuracy consensus reads. So for each of these samples, we obtained little more than 4 million single cDNA molecules. And so I think that's typical for this platform. And this recapitulates some of the results that Liz presented, the Alzheimer's brain and the universal RNA samples that have been publicly released. In particular, out of all of these CCS reads, we obtained about 70 to 80% of the reads being fully productive, in that we found both adapters and it's considered a full-length read. So I mentioned earlier that we multiplexed our samples, and so after the PacBio run, when we demultiplex the reads, you can get a sense of how much coverage you get for any one sample. So here I'm showing you the breast cancer 5 different cell lines, and on average I think we get very consistent pooling. This is something that happens upstream, and so it really is based on the method you use for quantifying the cDNA amount per sample. But overall, for the breast cancer set, we obtained about 1 million full-length reads. And then for the melanoma samples, we also get adequate coverage. I do want to mention that among these samples, there were 2 melanocyte samples, which come from patient skin biopsies, and then the melanocytes need to be purified out. So through this process, we actually obtained a very small amount of RNA, which is one of the explanations for the lower coverage for the melanocyte samples. But overall, this indicates that when you're thinking about your experiments, you can really pool anywhere from 2 up to 100 samples, depending on your needs. So with this data, we're detecting a wide range of isoform diversity. This is an example of the Scottie Output where we can get a snapshot of the isoform annotations and classifications. So as you can see, for the reports for the breast and melanoma lines, we obtained about 14,000 unique genes and over 100,000 unique isoforms. If we look at a breakdown of the isoform categories, you can see that about 40,000 of the isoforms exactly match the annotations, in this case, the gen code annotations. An additional 40,000 are incomplete splice matches, meaning that they match a gen code transcript, but they appear to be a truncated form. And then another roughly 30% of isoforms are novel. So at this point, I want to mention that the numbers you get in terms of the breakdown of isoform classification is going to really depend on the sample you're analyzing. So Liz had mentioned before that there were many novel isoforms detected in the Alzheimer's brain samples. There's a lot more splice diversity in brain tissue, usually compared to epithelial tissue like here. And so you should interpret these numbers with those sample types in mind. Oh, one other thing I want to mention is that the splice junction classification is also very helpful to look at. That is looking at the number of junctions that are canonical versus non-canonical. Most of the time, junctions are expected to be canonical, at least 99.9% of all the junctions. And so the fact that a majority of all junctions found from our de novo sequence reads are canonical is a very good indication. And this is a property that you can use to filter out isoforms as you wish. So we have found many isoforms per gene. And so another helpful thing to look at is the number of isoforms per gene across the samples. So to give you a baseline, I'm showing you the gen code basic annotations of all protein coding isoforms. And you can see that the breakdown over here, where approximately 2/3 of all genes have annotated multiple isoforms. In the case of our samples, there's a similar distribution, but we do find highly diverse genes that are producing many more than 6 isoforms. And so when looking at this data, I think this is really common. So when looking at gene annotations, you have to consider that it's a set of isoforms that have been curated. And so a lot of microvariability or disease-specific isoforms usually are not included in these annotations. And so in real samples, usually you find a lot more diversity than as compared to the annotations. Another point I want to make is that given improvements in the sampling across many different transcript lengths, including very long transcripts, and I think this is through improvements in the chemistry and the flow cell configuration of the new sequel-T system, without having to do fractionation, I think we're capturing a much wider range of lengths. So again, to give you a baseline, I'm showing you the gen code 30 annotations, so including both non-coding and protein coding transcripts and what the length distribution is from that. And then in comparison are 2 samples. And so what you can see is that for the really long transcripts above 10 kb, it's still a challenge to detect them, and that has something to do with also just the biochemical sample prep that happens upstream. But overall, we're really detecting a much larger range. You can see that we're detecting cDNA molecules up to 6 and 7 kb. So that's really promising. And then again, I want to mention that no fractionation has been done. And so if you really are interested in the ultra high length transcripts, you could even consider fractionating or doing an additional run in which you remove the shorter molecules. So given that we have many more reads per sequencing cell, I think we're starting to reach near saturation for complex transcriptomes. So here what I'm showing you is a saturation discovery curve in which we subsample the reads from our data set, and we ask the question, how many genes and how many isoforms do we recover given this subset of our data? And so what you can see is that for the gene detection, it plateaus relatively quickly, and that makes sense because all you need is a single isoform to detect a gene. Conversely, the isoform detection, that curve is a little, it doesn't plateau as soon, obviously because there are lower abundance isoforms that require more read depth to detect, but you can see that that curve is starting to plateau. And my guess is that with an additional flow cell, so basically with 2 ADAM chip flow cells, I think you'd really be able to capture most of the isoform complexity within a human sample. So here I'm showing you the breakdown of the isoform detection based on the category. And so it's a very subtle effect, but you might notice that for known isoforms, which tend to be higher in abundance, the saturation discovery curve tends to plateau a little sooner than the novel isoforms, which tend to be lower in abundance. But of course, this will be very sample dependent, but this can kind of give you an indication of how much more sequencing depth you would need if you want to really capture the whole extent of isoform diversity. Okay, so I've shown you quite a bit of data giving you an indication of the coverage. And I think a really interesting application is isoform quantification. I think this is a really important goal for the field, and to really know how many copies of each transcript is expressed in the cell will really open up a lot of avenues in biomedical research, such as having consistent biomarkers, understanding disease mechanisms, and even just fundamental biological understanding. So many of you probably know that most of the isoform quantification being done has been using short-read RNA-seq data, and it's been a challenge. So gene quantification is quite reliable using short-read data, but when you need to deconvolute very complex isoform structures, things kind of break down. And that's really exemplified by this study done by the RGASP initiative in 2013. I'll say that the situation is still the same, that regardless of what isoform reconstruction method you use or isoform quantification routine, the answers you get will be very different. And one of the reasons is that these methods are heavily dependent on having accurate transcript models. And so that's really the key, is that if most of the time we are actually, in every situation, we actually don't know the set of isoforms, a gold standard set of isoforms that are expressed in any given sample. And these models are crucial for accurate isoform quantification. So this is illustrated in a very nice study from Anna Kness's lab, in which they did isoform quantification under 2 different assumptions. So the first assumption was using RefSeq models, as shown in red. So given the assumption that you have 2 of these RefSeq isoforms, the conclusion made was that the second isoform was expressed on a much higher level than the first isoform. So then they actually had sample-specific PacBio data, and within that data, they showed that there's only a single isoform as shown in green, which has a shorter 3' UTR. So based on that correct model, they showed that it was actually a variant of the first isoform that was expressed at a higher level, and this was confirmed by RT-PCR experiments. So I'm really just sharing this as an open possibility that maybe with the increased depth and the lower bias in sampling of the full-length reads, that we may be approaching a threshold where we can start to think about isoform level quantification. Furthermore, a study done by Dana Wyman with the ENCODE 4 consortium, they did a very nice study in which they compared PacBio sequencing and Oxford Nanopore as well as short read sequencing. And what they showed is that the technical reproducibility of long read sequencing is very high. So here's an example in which they sequenced an ENCODE cell line, and so you can see that the two replicates are quite highly correlated. And then to the right is the same plot, but it's broken down by what the category of the isoform is. And so again, with this theme that the known isoforms tend to be higher in abundance, and so their quantifiability will have much higher precision. The lower abundance isoforms will tend to have higher variability. So that's a consideration in thinking about quantitative applications. And so we just did a little bit of exploration to on this question. So we took the same samples that had been sequenced on the Sequel-2 system, and we collected Illumina sequencing data on them, and we've started to compare these results. And so what we find is that the gene level quantification is very well correlated. So I'm showing you data here for 2 of the breast cancer cell lines. On the x-axis is the Iso-Seq transcripts per million. So this is full-length reads per every million reads. And then on the y-axis is the short read transcripts per million value. And so what we were actually surprised to find a very high Pearson correlation coefficient, especially given the way this data had looked with the previous platform. So I think this is very promising. We also looked at isoform expression, and I think this is more of an open question. So we find moderate correlation between the long read and short read results, but at the same time, there are some discrepancies. I'm going to give you a couple ideas of of how this could occur. So one example is that if there is an isoform that is extremely long, let's say more than 10 kb, and is not efficiently sampled using the long read method, it can be efficiently detected using short read methods because the transcripts, of course, get fragmented before sequencing. So that could be one reason for a discrepancy. Another reason we're finding that there's a discrepancy is cases where the short read isoform quantification routine kind of breaks down. So if you have a very, very high complexity gene and there are many, many isoforms, sometimes the algorithms don't do a good job of estimating the amounts, basically of apportioning the reads correctly. So 2 examples here. Obviously, there could be many other issues, and I think this is kind of an open, basically an area ripe for more development. Okay, so now I want to move to some experimental design considerations. Namely, when you're thinking about your experiment, whether to go with a targeted approach or a bulk sequencing approach. So recently, there have been many methods published demonstrating that targeted long-read sequencing is a possibility. So in terms of targeted RNA sequencing, I do want to give credit to Tim Mercer and John Wynn's group. They were the first to really demonstrate that targeted enrichment of RNA is possible within the application of next generation sequencing. But then a couple years later, the Lagarde group, Roy Johnson, and Roderick Guigo's group, as part of the GenCode consortium, really demonstrated that you can enrich for full-length cDNAs. And they did this by targeting link RNAs in human and mouse as a part of a gene annotation effort. And this was highly successful. Near that same time, Tim Mercer's group had targeted all the loci in chromosome 21, showing both link RNA and protein coding isoform diversity. So what I want to mention is that I've recently developed this method called ORF-Capture-Seq, which I believe stands as another complementary option for the previous workflows. So one thing I want to mention is that sometimes it's difficult or it takes a lot of resource to obtain the probes to do a targeted sequencing run. So that was one of the motivations to develop a versatile option to make capture probes really easily made. If you have available clones or a PCR product, you can make probes within a day using low-cost molecular biology reagents. And second point is that sometimes the sets of genes targeted may be variable. So depending on what pathways you're interested in, you may not want just a single complex probe set, but many, many different probe sets with different gene compositions just for flexible experimental design. And so this method is very good for that. So we demonstrated in this paper on bioRxiv that these ORF-Capture-Seq probes are comparable to commercial probes. So here we are comparing the, we actually looked at 3 low abundance transcription factors and 2 high abundance housekeeping genes as a control. And so you can see that without capture, you're mostly sequencing the high abundance housekeeping genes, but then after hybridization enrichment, a majority of the sample are the 3 transcription factors we targeted. And so again, I want to say that the complexity of the probe sets can be increased dramatically. So you can target anywhere from 1 to 700 or a couple thousand genes. And so here I'm showing you that the more genes you target, the more isoforms you detect. But given the same amount of sequencing, you will actually recover fewer isoforms per gene on average. And this has everything to do with the relationship between number of genes targeted and coverage. So, so here I'm showing you saturation discovery curves in which we've targeted two transcription factors, 12, 88, all the way up to about 700. And so if you're only targeting two genes, you need very few reads to reach saturation. Here we're seeing about only 10,000 full-length reads. Whereas if you're targeting 700 genes, you may need many more reads. I do want to say that when we did this study, we were using the RS2 platform, and now that the throughput is much increased, I think it's quite accessible to target up to 1,000 genes and really get full coverage on a single cell. With those thoughts in mind, some experimental design considerations will involve thinking about how many samples you have and how many genes there are of interest. And so the first situation is if you have very few samples and very few genes of interest, then targeted sequencing might be a good choice. Here's an example in which isoforms of the SNCA gene, which is relevant in neurodegenerative diseases, were targeted from different patient cohorts. And because you were targeting that gene, it was trivial to enumerate all of the different isoforms that were associated with normal versus the Parkinson's disease patients. Another application where you have very few samples but many targets. Again, I mentioned gene annotation projects. So in this study, a couple different tissues from human and mouse were interrogated, but they went after all predicted and known link RNAs. So in that case, you could get very, very high coverage across many, many genes through the targeted approach. And then likewise with our projects, we're very interested in discovering isoforms and then asking what their functional effect is. So this involves cloning the different protein isoforms for characterization. And so we wanted a broad map of what was expressed before doing these, applying these cloning approaches. On the other end of the spectrum in terms of experimental design are cases where there are many, many samples and few genes of interest. And really, I have not seen an application, but my guess is that this would be relevant to complex disease research, where the goal is to measure small effect sizes of isoform expression, or for biomarker discovery and validation. So I think these are two applications in which you want many, many individuals to really extract out or distill an isoform specific expression profile. So here I'm leaving you with some thoughts, and I'm very happy to answer any questions that you may have during the Q&A session. Okay, so with that, I described to you our experiences with the new Sequel II system. I think overall we're kind of reaching this critical point where, because of the throughput, we're able to capture much more of the transcriptome. I think there is a lot of potential to develop isoform quantification applications, and then I've left you with some ideas for experimental design considerations. So just a quick last point is a lot of us are doing quite a bit of sequencing and enumerating many, many isoforms. Many of the isoforms are novel, but I think a very important question is, what is their functionality? Are they actually affecting phenotype? And so within the center here at the Center for Cancer Systems Biology, we've developed a new approach to this question, and it involves identifying large numbers of isoform pairs for large numbers of genes, and then we are able to express the protein isoforms and ask what their functional differences are in terms of protein-protein interactions, protein-DNA interactions, or any other functional property. Because I think ultimately, when we're looking at this data, an isoform will matter, within a patient context or a biological context if it's affecting an interaction or some sort of biochemical function. And so overall, we've looked at this question on a proteome scale view, and we found that many isoforms do affect protein interactions. And so if you are interested in learning more, you can look at our paper in Cell. And then in general, I think overall there should be an empirical framework to study isoform function. So what we've been talking about a lot here is the isoform atlas, but I think in many of our projects, we're going to need to integrate this atlas with developing tools to manipulate the isoforms either through upregulation or downregulation, and assess their phenotype and then mechanisms. So this is an area that I'm very interested in. So please contact me if you for potential collaborations. And with that, I want to acknowledge my postdoctoral advisor, Dr. Mark Vidal, who's the director of our center. And I want to acknowledge Kate Tuttle. She's a student who graduated from Northeastern University recently. Very excellent student who really did a bulk of the development in targeted long-read sequencing, and she's currently looking for employment. So if anybody knows of someone, please definitely contact me. I also want to thank the sequencing service from the Maryland Genomics core at the University of Maryland, and in particular Luke and Lisa. So they did an excellent job in processing our samples. And then I want to acknowledge my funding from the Melanoma Research Foundation. And then the last thing I want to say is that I am on the academic job market and looking for faculty positions. And so if you know of any opportunities, please don't hesitate to contact me. All right, and I think that's it. Thank you for everyone on the line for listening.
Great, thank you, Gloria, and thank you, Liz. Just a reminder to our attendees, if you do have any questions for Liz or Gloria, please pop them in the questions panel that's on your attendee dashboard. And we do have a few questions to get us going. And the first question goes to you, Liz, and the question is, excuse me, imagine that there are two transcripts where one of them is an exact shorter version of the longer one. Only the short one is expressed and the read can map equally to both of the transcripts. How does Squanti2 redistribute the reads, counting both, randomly choosing one, or removing such reads?
Hey, Donald, can you hear me?
Yes.
Okay, yeah, so I can answer this question in more detail, but actually, I'll just put it this way: there's some confusion here as to what Sconti2 is doing. Let's say if it seems like the case being presented here is just like, in actuality there are two transcripts and one is the exact shorter version, but only the shorter one is expressed. So Iso-Seq would only get whatever's expressed. So Iso-Seq would only produce the shorter transcript. Now, then when you map it back to the genome and then compare it with the annotation, the question I actually have for the person who presented this question is, what does the database have? Because if you look at the GenCode database, I do not think GenCode actually annotates transcripts in which one of them is the exact copy of the other but shorter. So I think what's going to happen is the GenCode database is going to have only the longer version, and then you're going to map the Iso-Seq transcript, which is the shorter version, to the longer version in GenCode, and it will be categorized as an incomplete splice match or an ISM. Right. So I think this is a database thing, because databases usually do not want to maintain redundant transcripts that are just shorter versions of each other. If for some reason this transcript database actually has both versions, then Sconti's classification method always just assigns the one that has the best classification. So since FSM is a better match than ISM, suppose for some reason your transcript annotation actually has both the short and the long one, then Sconti would assign the Iso-Seq transcript to the shorter one, and that would be presented as an FSM. And again, if you have any questions with Sconti, I'm very happy to answer them. You can always go to the Sconti GitHub and open an issue and ask me questions over there as well. And the Sconti2 GitHub page also has lots of documentation on what all the different classifications mean.
Great, thank you. And a follow-up question for you, Liz.
I actually think I want to add one point to the annotation question, is that sometimes if a transcript is a subset of another, and if there's evidence that that second shorter transcript is valid, usually by evidence that there's a promoter there and it's active, then I know that that situation, it does get captured in the annotation, but I think it's rare.
Yeah, and actually, Gloria, this reminds me, we should also have, so people were asking about the 5' end, but you and I have actually talked a lot about the 3' end, right? So we've talked with the GenCode people about the fact that usually there's only one, they usually have to pick, even though they know there's multiple polyadenylation sites for the same gene or transcript, they often will only pick something that is either the longest or they think has the strongest signal.
Yeah,
so
I was alluding to that point too with, so in the Iso-Seq data, we find many isoforms, but usually in the annotation, they try to be parsimonious and only pick like a representative isoform if there are many isoforms that only differ by each other, by a nucleotide or so. So yeah, I totally agree with that point.
Next question for you, Liz, is can you do single cell sequencing using Iso-Seq?
Yes, absolutely. So for this particular webinar, I did not mention on single cell, but we have actually already been doing some single cell data. If you're interested, if you go on YouTube, type in AGBT, Jason Underwood, there's actually an AGBT talk this year on applying Iso-Seq to the Dolomite Nadia drop-seq system. And there's also been a publication by Hagen Tilgner using the 10x system with Iso-Seq for single cell. And yes, the one of the great things about the long accurate is that we need minimal or no error correction to match these barcodes back to matching Illumina datasets.
Thank you. And a question for you, Gloria. Is there a protocol available for the ORF capture-seq?
There is. So all of our work is on bioRxiv. So if you do a search and look in the methods, everything, all the details should be there. If there is something, more information that is needed, feel free to just email me.
Great. And actually, just in terms of emailing you, just if you could pop your final slide up into slide view mode so people can capture your email, that would be great. Thanks. So moving on, another question for you, Liz, is using human as an example. What is the number of reads per sample you should get target for a typical standard Iso-Seq project?
So this is a really good question because as you can see, both Gloria and I presented a rarefaction curve, so sampling efforts on both the UHR and the breast cancer cell line, and also the Talon paper by the ENCODE 4 project has also done some sampling efforts. I think we estimate that at about 1 to 2 million reads, we've already captured 80 to 90% of all the genes that we could capture. So I think 1 to 2 million reads is quite sufficient. It really depends on the complexity of the sample. You know, Gloria was sequencing a cancer cell line. The UHR is also a mixed bags of different samples. So, for example, if something is more simplistic, I'll just put liver out there, it's possible to sequence with even less.
Okay. And another question for you, Liz, is and this is kind of a compute question, can Iso-Seq data analysis or SCOANTI2 be run, in parentheses, albeit slowly on a laptop, or does it need more significant compute power?
Actually, Scottie, there's two questions. So SCOANTI2 is actually a very lightweight program. It absolutely could be run on a single laptop. For Iso-Seq, I have actually not tried this myself, but I would say you could give it a shot. One thing that might benefit is if you go to the PacBio GitHub page for our Bioconda releases, because the Bioconda toolkit is a lot more lightweight than our SmartLink/Smart Analysis toolkit. This is a question that, again, if you go to the PacBio software pages or GitHub pages, I think you'll get a lot more answers on what the computing requirements are.
Thank you. And another question directed at you, Liz, is does Iso-Seq match short reads for quantification?
So this is an open question, and I'm also going to let Gloria comment on this. The question is, does Iso-Seq match short reads for quantification? Gloria has actually just shown a matching sample of the cancer cell lines for both short and long read data. I'm also looking into this. I think on the gene level quantification, the short reads and long reads do match quite well. On the isoform part, I think it is really a mixture of short reads are simply not possible to quantify accurately at the isoform level. Plus, a lot of the tools that we're using, like RSEM and Callisto, are also not designed to understand full-length isoforms. So it's really hard.
I think I'll jump in there. So the tools like RSEM, Callisto, Sailfish, they do a great job within the assumptions of the method, which is they assume that the gene model is correct, that the set of transcripts you give them are the set of transcripts that are in the sample. Where it breaks down is when there's an isoform that's in the sample that's not reflected in those models. So I think that's a really big challenge. The challenge is ultimately that we don't have that gold standard. We're actually still figuring out the parts list of I don't think there's a single sample where we know the identity of all the transcripts for that particular sample. And that's what we need for short read quantification.
But I'm also going to argue that if you have a very complex isoform, like 10 or 100 isoforms from the really complex neural genes, it is just simply not possible to accurately assign every short read. Like, it will become a probabilistic problem. And that's why I think there is some desire in the community to use long reads solo for quantification, or at least relying a lot more on the long reads for quantification.
Mm-hmm. Yeah, I agree on that point. You're talking about the non-deterministic. Sometimes just having short reads alone is not enough information to accurately quantify isoforms.
Okay, well, we do have a couple of more minutes for questions. If anybody wants to ask some questions at the tail end of this broadcast. There are no other questions at this point. Actually, here's one. How many 1M cell samples would you suggest starting with if you only have a Sequel 1 system?
So on the PacBio Sequel 1 system, there's 1 million ZMWs. However, usually we target 500,000 full-length reads. This means that for each SMRT cell on the Sequel 1 system, you'd expect 500K reads. And I had previously answered that I think you need probably a minimum of 1 million reads to start seeing quite well all the genes in the whole transcriptome. So I would say if you have a Sequel 1 system, you might have to start with 2 SMRT cells.
Okay. While we'll wait for any late-breaking questions here, I will just quickly add some closing comments. So first of all, just to mention to everybody, we have recorded this webinar. We'll have it available for download in the next several days, so keep an eye out for a follow-up email with a link to the recording. Second, our Human Genetics SMRT grant is currently open. So if you have an interesting research project that could benefit from SMRT sequencing, consider applying to win free sequencing. And we are accepting submissions through Friday, November 22nd. And for more information on this specific SMRT grant, you can visit pacb.com/smartgrant. That's pacb.com/SMRT And finally, to keep up to date on our full portfolio of webinars, you can visit or bookmark pacb.com/events. So I'm just going to see if there are other questions. So yeah, there are a couple of more questions that we can fit in before we end. So
yeah, this is Liz. I can see the questions.
Okay.
And actually you've already, because I didn't know we had answered questions had time, so I actually quickly answered them in private, but I'll just quickly answer. For one question was linking isoform expression to epigenetic states. There is a recent publication looking at methylation, and that is the Iso-Seq paper. I recommend everybody take a look at it. However, I would say on the PacBio side, we have done not much to look at this, so it's an open question. The second question was what coverage would be suggested for isoform quantification. This is also an open question. I think the data set that we've just released on the Alzheimer and the UHR data set is really a starting point for people to look at this. I'm really invested in releasing public data sets to motivate the community to look into some of these more nuanced questions. And the last one is it possible to sequence RNA other than mRNA, such as ribosomal RNA? Well, anything that has a poly A tail is possible. And if you don't have a poly A tail, then the protocol will need to be modified. There is actually a Nature Communications paper out recently on doing bacterial Iso-Seq, which does not have a poly A tail, and they did have to modify the protocol.
Great. One other last comment. Liz mentioned our colleague Jason Underwood in an earlier answer to a question, and just wanted to mention that he will be talking in a webinar with Dolomite Bio on this December 3rd on single cell isoform sequencing. So I'm sure if you go to the Dana-Farber Bio website, you'll be able to find a link to registration for that webinar. So please check that out. So that's all we have time for this morning. I really want to thank Liz and Gloria for sharing their expertise as well as their time. And again, please keep up to date on our other webinars coming up at pacb.com/events. And with that, we'll end this, and thank you for joining us, and we hope to see you on the next webinar. Take care. Have a great day. Bye-bye.